We are live now, sir.
Welcome, everybody. I hope you all had a great Sunday. This is part of our continuous integrative medicine case report, continuous education series. Today, we are joined by Dr. Arunan, who is one of the famous educationists and also a distinguished member of the Indian Academy of Neurosciences. He works at the Tata Institute of Fundamental Research, Mumbai. He has been associated with the Homi Bhabha Center for Science Education. And he, along with Mayur Gaikward, who is an assistant professor from the Alfinston College, Mumbai, has been associated with developing the CUBE model, or the collaborative undergraduate biology education model. It's now called collaborative understanding of biology education instead of undergraduate, because of the wide expansion of the entire model to across the country, the students, the high school students, the college students, and they've been doing experiments of all kinds. And today, the planned event had Dr. Arunan talk about the way we do different experiments. And when we fail, we should get up and do them again without being fearful. And the topic of today's discussion is cultivating science through celebration of goof-ups, which implies that we must continue to work and experiment even if we fail. And this applies to all kinds of research in our integrative medicine research, the modern medicine related research, the drug development programs and the failures and the success of it. It can apply to the Ayush research when there are experiments which are going good and there are those evidence that needs to be disregarded or to be translated for social good. So unfortunately, Dr. Arunan couldn't join us because he had some trouble trying to come live. Although every day 3 hours he's trying to connect with various students of at the bachelor's level and educate them about how to do experiments at home because they are not able to come to his lab. So we thought this will be a good idea to request Mayur Gaikward instead to come and introduce about CUBE, his relationship with Dr. Arunan as a mentor and as his a person who is developing this CUBE program. Dr. Mayur is, as I mentioned, the assistant professor at the local Elphinstone College at Mumbai. So, Dr. Mayur, why don't you share your experiences of working at CUBE and working with Dr. Arunan? Over to you.
Yes. Thank you very much, Akshay. So we have started Q program actively from 2012, and I guess I was the first student there as a first year BSc, and from that we have developed uh, lengths and breadths of the country and even abroad. And so earlier we used to call it collaborative undergraduate biology education, but when we found that even high school students, postgraduates, graduates are also need to be included in this program. So we thought of changing it to understanding biology education. So this is the crux of the program. So I will be going through the some presentation over here, which me and Arunan prepared, and so I will take you to the journey of how what are we exactly doing and what is our objective in this whole program. So the question here, what these are the some earlier slides which are prepared by the Arunan himself. So the question what we are asking is, where are the spectators in science in India? For example, so can we identify uh, this this person over here, this gentleman over here? So when we do this program anywhere, so the most of the audience will not have an answer to it. And if we go one step further, even this person, do we know this person? So still there will be a silent. And I guess if the people who are watching online, they can also think that who this person is. And if I go one slide further, then this person, so if I show this person, then everyone will understand and everyone will know who this person is. He is the our Indian cricket team's captain, Virat Kohli. Yeah. So what is the problem here? So the thing is, why we know Virat Kohli and why we don't know who the earlier chap is. So why we know Virat Kohli is because of the, okay, I guess Arunan sir is coming alive.
Yes, welcome, Dr. Arunan. Mr. Mayur Gaikward is taking on the presentation, and we will come to you shortly. Yeah, Mayur, please carry on.
Yeah. So the thing here is why we know Virat Kohli very well, because each and every person in the India knows Virat Kohli, because each and every person in India plays cricket very well. And there are some and hello.
Yeah, you're fine. You continue, please.
Okay. Sorry.
Dr. Arunan will shortly join us now that you've got started. He's
Yeah.
fixing his camera. You can carry on with very nice presentation.
Yeah. Okay. So just a minute. I'm just trying to get the slide. Yeah, I'm very sorry for this. Yeah. So what we are speaking here about the Virat Kohli, why we know him, because we play cricket very well. And the earlier chap which we were talking about was was Venki Ramakrishnan. So Venki Ramakrishnan is a Nobel laureate who worked in who actually did his graduation and his PhD in physics, and who got a Nobel Prize in chemistry working on the problem of biology. In that was the enzyme which he worked on. So why there is a problem in not identifying Venki Ramakrishnan and identifying Virat Kohli? Because there are no spectators for science. There are no people who understand science. So so we are asking here, we are proposing here a different model, that can Indian science learn from the Indian street cricket? Because uh, so the thing is, where are involved spectators of science? Here we can see the very nice picture where everyone is watching playing cricket, because everyone understands cricket very well. Then everyone knows the rules of the game. And if there are some rules we want to tweak and twist, we can do that. There are no complaints. For example, we don't have stump, we can keep one wooden flock or a tree or anything, so that people can play. If we don't have bat, we can keep some wooden thing and we can play. We don't have ball, we make up some ball and we play. We don't need season ball for playing. Then if again the same thing, if you don't have stump, we improvise something here. Then so every nooks and corners you will see, you don't need a very good and fancy ground, you can play this cricket anywhere. And you get a feeling of a cricketer, that you are a cricketer like Virat Kohli, Sachin Tendulkar, etc. And nothing can stop in this pursuit. Whatever you do, the people will play cricket, people will understand cricket, people will get enthusiastic because of this cricket. And what are we saying this, the rules of the games near the street are the same, and perhaps there is more fun, we laugh, we cry when we are playing, than what the normal people, or than what Virat Kohli or Sachin Tendulkar must be having. So as I was saying that the rare we, we don't understand, we don't recognize Venki Ramakrishnan. First of all, we don't have that many Nobel Prizes here in India. We have very handful of them, and one of them is Venki Ramakrishnan, but we still don't recognize him. So what are we proposing here is, we are proposing here is the STEM model, the gully cricket model. So can we do science in gully? Can we do science like how we play gully cricket? So this is our cube lab in HBCSE, HBCSE TIFR. And on the door it says that no meek, no weak, no geek. There is no selection. Everyone is welcome. So it is very inclusive program. Anyone can join. And so cube. So we also say, so earlier on only I told you about there's a vertical integration of school colleges. In this picture you can see different ages, different professions, different looking like people are coming together and celebrating it. And this is the real power of cube. And we started from 3 centers in Mumbai, 3 colleges in Mumbai, and now we have reached far and breadth, from Jammu Kashmir to Tiruvananthapuram, from Assam Sakepal to Ahmedabad. So all the corners we are covering. So I guess Arunan will like to go upon these slides.
Dr. Arunan is not there, so you can.
Okay, so what I will do, I will go a little bit ahead. Yeah. Yeah, so what I was talking about, the gully cricket model. So I will give you some examples of this gully cricket model, how we do the science in our labs and everyday life. So what happens is a very simple activity like trapping the fruit fly. Now, fruit fly, Drosophila melanogaster, people are seeing, people are learning, students are learning in the textbooks, in their journals, and but we don't connect this Drosophila melanogaster with the fly which is coming to our kitchen on our fruits. So to create a little bit awareness, we started with this program, the Trap Your Fly. And so what happens is students started trapping those flies. School students were there, college students were there, postgraduate students were there. So earlier they were not getting flies. So at every day in our cube lab, what we do is, or anywhere you are there, people do some experiments, people, and they report in evening. They report via discussion, they report via WhatsApp, they report via STEM game app. So there are many facilities and there are many easy resources which we can communicate very easily. So we asked students to trap some flies, and student did that, and they were not getting the flies. So in the discussions, we started asking that why you're not getting the fly. And the students were saying nahi aata hai bhaiya, flies abhi kaise aayega. And there are some students were getting, some were not getting, and there was classical reasons that flies are not coming. Then we then they some of the students started getting flies more number or in less number during some day times and night times and something like that. So we, while discussing, we said that why don't we start observing these flies every after 2 hours. So what we can do, we can keep a normal bottle, keep a banana peel, and just keep the this trap in near our kitchen dustbin and see how many flies comes. Very interestingly, the student recorded this, and what they found is very astonishing. That in a daytime, so they used to start from 7 a.m. 7 a.m. till evening 7 p.m. So they found that they were doing this in December, and they found that majority of flies are coming around 11 to 1, and in evening 3 to 5. So and this was in December, and when they and we started calling it activity pattern. So there is some pattern in this. And when they did the same experiment in, let's say, June, the scenario was totally different. Like in June, they got majority of flies earlier than what they used to get in December. So they got majority flies on around uh, around 7 to 9 a.m. And in the evening, they got 3 to 5 p.m. So there was heavy shift in the amount of flies they were getting in if you talk about time. And and then we started reading about this, why this is happening, and the students connected this with the sunrise, that in December the sunrise is late, that's why the flies are coming a little bit later, and in June the sunrise is earlier, that's why the flies is coming earlier. And then it's like a hell broke loose. Everyone started reading about what is this concept, so we started asking questions on activity rhythm and circadian rhythm and stuff like that. And it was amazing that the students started hypothesizing that when when the maximum number of flies you will get at what time in what part of our country in different latitudes and longitudes. And so people started learning, so people started seeing the literature about, do we have any literatures. We found that there are very classical studies from Konopka and the group which were talking about, there are some genes, there are some proteins which are required to maintain this behavior, this rhythm of activity. And it is very interesting that in 2017 the collaborator of Konopka got the Nobel Prize, and everyone was excited because the students were yelling that we got the Nobel Prize, because the Nobel Prize what they got was in their area, was on circadian rhythm. And this was very exciting. So they started feeling for this. And this all came because of many goof-ups they did, many mistakes they did. And then we discussed, they celebrated this, and this is the result of that. So this is a very classical example how it happened. So what can trapping? So the simple activity like trapping can can open the big box of questioning and misconceptions and different conceptual gaps. For example, so in discussions we normally ask, so why do the fly come to the banana? So the people will say because they smell. So someone will ask, what do you mean by smell? So the concept of smell itself is a very enlightening concept, that okay, smell means like aroma or what. So then people, okay, smell must be made of something. So then some students will say, okay, chemicals or some molecules. So then the concept of diffusion, so the concept of molecules, so these concepts come into the picture, into the lamplight. And how do we smell? it has a huge neural it has a huge neural background. People, the students should know the structure of neuron. And this all we kind of ignore while we were in high school, and in our undergraduates, most of the postgraduates also, we kind of ignore the structure of neuron, the how the neuron carry impulses and stuff like that. So this becomes a very interesting opportunity for us to discuss, because the examples are very alive. And this is something which what happened, that of the demystifying concepts of lab. So here we can see in Binita, in some 2 3 years ago, what happened, the lab was closed, so she started culturing drosophila in their home. And home means culturing drosophila home is to make medium and stuff. So she used to bring the contents from the lab, some contents from the lab and some contents from the home, like sugar, sucrose, she can take sugar, yeast extract she can buy or something like that. And autoclave was not there, so she started working with the pressure cooker. And so there was a very new thing which happened, that people can study this thing, people can work in their homes. And this whole demystify the concept of lab, how lab should be. What we talked about earlier, the gully cricket model, it is very much applied over here. So we can work with anything around what we have. The second thing which happened during this lockdown was that the people were again wanted to work daily. And we asked students to make some media, we asked students to trap some flies, they trapped some flies. The thing was they wanted to culture those flies. So how to culture those flies? So for the culture you need media, and the media, the component you need sucrose, dextrose, corn, agar. So the thing was how to do this. So a group from Kerala and a different part from Mumbai and different, they collaborated, and they came with their own media. They found the substitution of agar, they found the substitution of maize powder, they found the substitution of dextrose, and they invented this new kind of medium called rava tomato vinegar medium. And what happened with that is, and this didn't happen overnight. This we did like discussions months after months, like for this whole lockdown period, daily with hit and trial. We Now we have a very good understanding of this media. Now the media is very good, media is working. And this is how we came. So this in this media making, there was a lot of group of which people came forward, they discussed, and it got sorted. And this is how now it is kind of revolutionizing the whole media scenario. Now people can culture the Drosophila at the home with the homemade media, and they don't have to go in the fancy labs and stuff like that.
Thank you, Mayur, for that very interesting way of popularizing science. And you mentioned how why the cricketers are more popular than the Nobel Prize winners. You showed a nice picture of Enki, who's a Nobel Prize winner. While Dr. Arunan was trying to get in, you nicely also explained how important it is for us to continue with our science even though lockdown is there. And you described very beautifully how you replace the general regular media you required for Drosophila by the homemade therapies, homemade culture media. Very nicely explained. And this is also true for people who have been locked down in whatever spheres of activity they have been involved in. They're trying to do their best and trying to innovate. And now we are joined by Dr. Arunan. He was supposed to give an opening talk. He's with us. We are very happy to have a very enthusiastic educationist and a dear colleague, distinguished member of Indian Academy of Neuroscience. There is an Indian Academy of Neuroscience in India, which comprises of about 1,400-1,500 members who do research in neuroscience from various angles, from the cell culture in vitro systems, from Drosophila, as Mayur just mentioned. In fact, we had Dr. Lakhotia from BHU other day. He's also from the Drosophila background. He uses Drosophila as a model to study. Some people use animal models to study and screen drugs, to validate therapies, and others also look at the other efficacies of various yoga and meditation modules to study the brain rewiring and using EEG and other systems. This is all science, and I will formally introduce you to our distinguished colleague, Dr. Arunan, who is a PhD from BITS Palani. He's also a Kishore Bharati fellow at the Cube Labs, as Mayur mentioned. He's one of the architects of this collaborative understanding of biology education, taking science to the colleges, the school students, even high school students, to engage them in curiosity-driven research, small things, just like he mentioned, how to innovate an agar media at home to culture Drosophila, because as I mentioned, they are very important models for neuroscience research. So he drives that program at the Homi Bhabha Center for Science Education, and also he has been associated with TIFR Mumbai, Tata Institute of Fundamental Research, and I've had the opportunity of visiting this Cube Lab at the Homi Bhabha Center of Science and Education. I was so thrilled to visit, and I was inspired myself. And then he visited our academy meeting in Chandigarh. And at that time, we were doing Clean India activities. The entire research people used to volunteer. And then one of those dustbins, we had a lot of Drosophila, and we went out there and discussed the Drosophila and its science while cleaning up the garbage outside PGI in some of the places outside. And this was a very good opportunity. Dr. Arunan has also headed the department at Life Sciences at the Sophia College for Women in Mumbai. So we are very privileged, and it's a proud privilege to have him on our platform and to present it, Dr. Arunan, to all of you. He will then now step in and take the presentation as it evolves. Over to Dr. Arunan. You need to unmute your mic, Dr. Arunan. Yeah.
Okay. So sorry for the goof up that I was not able to get connected. So it's in a way it's exactly that. This is exactly what is happening. Can you hear me? Can you hear me?
Yes, we can all hear you.
Okay. So this is exactly what we wanted to talk about. When there is a goof up, this of course I can't hide because everybody knows it that I was not able to connect with. But in the lab every day, in the lab when we do work or when we learn things, every day there will be some mistakes that is happening. But our education system and our society do not allow us to talk about it. So we hide it. And we hide that, I think students hide that when they are doing project everywhere. So one of our, I suppose that Mayur must have mentioned about it. So this goof up of that I was not able to connect with you people gave some great advantage that Mayur was taking over and have given probably better than what I would have given. I hope Mayur, you must have shown all the slides. I could not see that. Okay, so that's fine.
You can tell Mayur, you can, it's a very informal drawing room kind of a discussion where people learn from our discussion and interaction. You can brief him what we showed. I think he showed some cricket slides of Dhoni. I'm sorry, I'm very poor with cricket. Some leading cricketers, and he showed the Nobel Prize being Venki Raman's picture, and he made a point why it is not so popular. So let us make science as popular as cricket. But he did not elaborate how to make that happen. Maybe you can shed some light on that.
Yeah.
So I stopped here, sir, and from here you can take So I didn't explain. So I just gave the introduction of Cube.
Okay. So can you hear me? That okay.
Yes.
Okay. So this is the slide curious. Okay. So here we have this vertical integration in the sense all type of students you can see from 7th standard to postgraduates and all type of people. So this is what we would like, and Mayur must have mentioned about how great ideas come from 7th standard and 9th standard students when they mix with college students and others. And the other side of it is that the inhibition that generally college students have, because they expect that themselves should be knowing everything, will also vanish when they interact with, let's say, 7th standard and 9th standard students. So this has been one of our great experiences, and this we continue with that. And there emerge a lot of things. So what I was talking about this, the mistakes that one makes that we try to hide and not try to tell about will not happen, because some of the school students will say that no, he was doing something which happened like this. So there are examples like that. I think Mayur must have mentioned about trapping the fruit flies and looking at when do the fruit flies come in large numbers through the day and night, which students were doing along with their parents. School students were doing along with their parents, and so they get this data and they come and try to plot it. And I think that plot probably Mayur must have shown it. I don't know whether we have that plot. He must have shown it. And then when the, okay, no, I think I'll come back to that. So this, while doing this, there will be a lot of mistakes that will be made, and that will be exposed. And so this gives them a sort of culture where there is nothing wrong in accepting that that he or she committed a blunder the other day, and she or he got over that. That's the most important thing. And I will tell you one more example of that. We used to have, I don't know, we have a video of that. Yes, I think I will go find. Mayur, can you just show the video of the Moina?
Video, the photo you mean to say?
No, photo of the Moina. Yes. Yes. Okay. So this is a case of a freshwater small organism like a prawn, but it's only less than 1 millimeter size, which school and colleges, I think college students sometimes, biology students sometimes use it to call it as Daphnia, which is like a prawn-like organism, a crustacean. And that somebody got it from one of the colleges, some tank. There was water, and they were taking the water and bringing it. In fact, Mayur's college, when Mayur was a student, Mayur's college, I think somebody brought it, and when they looked at the water for algae, they found that the small organism growing. So it looked like this so-called Daphnia, which curriculum, syllabus, the practical syllabus asked people to do. So this was being, so then they wanted to culture that, and so normally how to culture, etc. Anyway, to cut it short, they were able to culture it with they wanted to culture it with the bacteria. So then they were discussing about E. coli, because what they knew about is E. coli, and so we said that okay, even if E. coli is not there, can't you give any other bacteria? So then at last they came up to when you keep some milk overnight, the milk will get spoiled, and that may be because of bacterial growth. So can we just put a drop of milk? So they were using a drop of milk, and then this organism started multiplying, and they got this a hell of a lot of organisms. Somebody started this, then they looked at it and found that some of these things can multiply from one parent without mating, parthenogenesis or something. So all these things came about. So they got a culture of that from one single organism. And so that means they have a stock from a single organism, which is a very interesting thing. And so when they got back to their colleges, the colleges and schools, they took it back. So the school students were to make this culture, maintain this culture. So that's the story that I wanted to do. So they were maintaining this culture. So the standard method that they have developed in the lab during the summer vacation, I think in 2012, was to add 1 drop of milk in 250 ml of ordinary tap water, which they kept it open to remove the chlorine. And so that water they were doing it, which they were doing it at home also. They tap water, they kept to keep it overnight and add approximately 250 ml in a bottle like this, water bottle, plastic bottle. And many students were, so the school students were told to make, each school to make 1 million daphnia. So they were making this, then Saturday or Sunday, they used to come to the lab and show this. So all of a sudden we found that one of the bottles, the so-called daphnia, looked very red. So that was, some of us were very curious about it and asked what happened, what did you do? So then this young boy said that, no, we did, I did the same thing. But another boy standing there, he said that, no, sir, when he was doing this, instead of adding 1 drop, he had added 3 drops or 4 drops, more drops fell. So that became an interesting issue to inquire about it. And then somebody started doing that with 3 drops of milk. And they found that this 3 drops of milk make this daphnia red. So which people looked at the references and found that the redness is due to hemoglobin. That's an astonishing thing for us. So there was a journal article from a journal of biochemistry, and found that daphnia produces more hemoglobin when in hypoxic condition. So that means more milk means it costs more bacteria to produce hypoxic condition. And so that was repeated, and has been people have been doing that. So hypoxia, so that was an issue that they were addressing now. What does hypoxic condition, how does hypoxic condition induce gene expression, hemoglobin gene expression? So that is an interesting story that took it to very, very exciting on a molecular basis of hemoglobin expression, and also epigenetic, possible epigenetic process of hemoglobin gene expression. But it didn't stop at this. How much time? How much time I have?
You have time, 10 minutes maybe.
Okay.
But I wanted to ask, when
yes.
the hemoglobin was expressed, you call it epigenetic, meaning that the milk acted as a outer influence to influence the gene expression of hemoglobin. Is that what you're trying to say?
Yes, I will come to that. So that what I'm trying to say is that so from very simple mistake like that, which the poor boy was not able to accept, the neighbor, the other boy sort of revealed it, and then from that came huge area of exploration, including an area called epigenetic, which is just touched upon in syllabus, even in BSc and MSc syllabus, and nobody really understands it. And here students started, from school students onwards, they started addressing questions like this, and they started seeing that what is epigenetics, what's the difference between genetics and what is epigenetics, etc., etc. So I will come to that, but there is another very interesting story in this, which I wanted to tell. So this is, of course, a story which says that from a small mistake, when it is sort of discussed with a peer group of different ages, a lot of interesting stuff like an epigenetics, idea of epigenetics come about, and exploration come about, and there are people working on that. But the real story is something like this photo. So one of this person who is studying and holding this, she was a young student there. Now she's a PhD student. And she took this, she is from Delhi, and she took her to Delhi. And when she was doing this, she wanted to address this epigenetic question, and she was doing that. But one day she wrote to me saying that, personally, she wrote to me saying that, sir, you should not get angry about it, but I found that what you people are talking about is not daphnia, it's another species called moina. Okay, so that was a bombshell that is thrown at us. This is what I wanted to tell you. So then we, of course, we didn't accept it. We explored it, and so she said that she has done it. She has done it, and she looked at it, and she also contacted some experts in freshwater biology, and she gave the names of those people, and she connected me with them. And she showed very clearly the reason why this is not the organism which is called daphnia, but it is moina. Because there are features, distinguishing features, which shows us that it's moina, and we could not deny the fact that she was talking about. Now the story didn't stop at that. I hope there are other. Mayur, could you just show the next slide, please? Yeah. So the story didn't stop there. Okay, this is some students took this when one of the students took this Moina when she finished her BSc and went to do MSc in Bombay University, Mumbai University, and she gave it. She took it there and she wanted to do address this question for epigenetics. But one of the teachers there, she got interested to find out. She wanted to do the molecular taxonomy of that. So she wanted to extract DNA and look at it on RNA, look at it and find out the molecular taxonomy. And for which she got the help of another scientist from the Bombay University campus, but from the atomic energy group called the CEPS. They did this. So this is actually the GenBank data. So they did, along with the students, they did this work and submitted in GenBank. So they identified it as a Moina macrocopa JSK1. Okay, that's the name that they had given. So this is the type of story that I wanted to tell, where things doesn't stop just like that. It goes on and on and on. Akshay, you wanted to ask anything, or
yeah, it's the nature of science, as you said, that once you hit on an observation, you define and characterize it, and if someone likes it, takes it forward. Like you showed, this girl went all the way to PhD and then challenged you that this was not Daphnia, which is another discovery, I would say. And then now someone has characterized further with this internal transcribe spacer too, which is probably another paper. So this is what I wanted to know is, in general, can you tell the audience why handling Drosophila and why culturing them, and then why describing its genetic structure is important for our lives? How does it, why should it inspire our own audience, which includes some of them are from the, of course, some physicians, families from the Ayush systems. How can we inspire them to take this as a cube model? Why should cube model affect their lives? Why should they, what message should they take for doing their activities to a new high? Why should they include research in their daily activities?
So I don't know whether I understood what you are asking about. If it is for fruit fly, I think probably Rakotia would have been the ideal person, Subhash Rakotia, and he was there yesterday with you. But what I can suggest you is that our idea is not only to restrict to fruit fly, but we have a very interesting stuff to do with fruit fly. I don't know whether Mayur mentioned about it. Fruit fly is, I don't know whether Mayur mentioned about that. I will repeat it. I think it's worth repeating it. It's fruit fly is a 6 times Nobel Prize winning model organism. I don't think that any other model organism must have been the reason, the cause for getting so many Nobel Prizes in so many different areas from 1920 onwards till last year, 2 years ago. Various areas, including immunology. Now, and this is the same fruit fly that is there in our kitchen, but we never connect this to. That's what I think Mayur probably must have when they showed the slide. Venki Ramakrishnan, it's very rarely that somebody gets a Nobel Prize, but we never rejoice it. We never even identify it. We never even connect it. So likewise, a school or college where they will be, of course, they will be teaching Drosophila in a very alienated way, without showing that this is the same fly that comes to your banana, ripe banana. Or now we found that even we never thought that it will become coming to even onion, it will come to lemon, all type of things, that if you, anything that is cut and open, I think it will come. So the point that we wanted to talk about is that as an example, taking fruit fly as an example, that the fruit fly is an alienated model, alienated subject when we are teaching and studying genetics and several other areas. Of course, as I mentioned about It's one of the frontier model systems with which people address all type of questions, including immunity, innate immunity. I think the Nobel Prize went to that, which nobody thinks about. So I think you said that about connecting human beings with the human aspects with this fly or any of these model systems. Now, right now, yesterday, last night, and till now, of course, our webinar is going on there in our group because it's around the week program. What I was talking about yesterday to today, the number of mails, number of messages coming to what we call it as chat shala is coming to on immune system is just triggered by the study of students studying catching, trapping fruit flies and culturing it and things like that, what Mayur was talking to you about. From that comes up this question, what all things are being done. So then now the people are asking a question, what about the innate immunity with respect to even COVID? Okay, COVID-19. Or what is there any acquired immunity possible to be inherited? There are, all of a sudden students come up with research papers which we never ever imagined to even access. Now we are forced to read that. Okay, I'm saying that we are forced to read that in the sense we are also very excited to read that. And not that reading is very easy to, reading and understanding is easy, but reading along with the students is easy because some of the ideas that we miss out, they will be coming up with their own ideas, which may not be completely sort of ripened stuff, but we can develop it. So this is what I would like to make a comment. So now what happens is that during this time, we also do it like street cricket. People started asking, what is it called in, let's say in Santali? What is it called in, what is a fruit fly called in Hindi? What's a fruit fly called in Haryana? What's a fruit fly called in Tamil or Telugu, etc. So people are coming up with this. Is I wanted to take this point. I don't know whether it is the point that you were asking about. So if you look at it, many people don't have a different name for fruit fly. Many languages, and they will say maki or that type of thing. So some have got, which will be small. They will say a small baby fly, something like that. That type of meaning. Now this, I wanted to take it to another level to say that, look at, that's fine, that, okay, this is an insignificant type of fly, so people are not bothered about it. It doesn't even cause probably larger amount of infection, so nobody is bothered about it. It's not a very relevant thing. So we were having a discussion on that, and so somebody said, no, what is relevant? People know about the features and they identify it. For example, snakes. Average person knows, in a villager knows that what type of snakes are poisonous, what type of, so they have a categorization of the snakes. But many of the other things they don't have. That's because it's not relevant. So this is very important thing because it's a sociology and sociology of science. So why categorization and classification is being done naturally, and why people like Darwin and all that started looking at these things and classifying, etc. This sort of questions come. The point I wanted to emphasize here is that look at butterflies. Okay, fruit flies are very insignificant. You can't even find that easily. But butterflies, we know that butterflies are so brilliant, beautiful butterflies are there. Some of them are drab. Plenty of butterflies we see it, but we don't have names for these butterflies. But whereas we had to take British names or the English names. So why this is happening is a question that we are addressing. And then we came to know that in Kerala, I think Kerala Shastra Sahitya Parishad and the groups which are doing butterfly studies, they started now giving names to local names to, or inventing local names and giving it to various butterflies. Now, this is very interesting. Wallace and Darwin before could identify plants and animals and look for evolution and evolutionary, find out about the evolutionary significance and come up with the evolutionary theory and stuff like that, is because they knew the different organisms. The classification was already done. So I think I will stop at this to say that we have a lot of things to look at to see that how we can popularize these organisms to see that everybody will be interested in, and then automatically people will start categorizing, classifying, finding out diagnostic features, type of thing. So at one end, I was talking about the molecular basis, etc., including epigenetic processes. The other side, we are discussing about very, very ordinary but very drab process of taxonomy. It's a very drab thing. If you ask any biology student, I think most of them will hate taxonomy, systematic biology and taxonomy. So, but the students who are involved in this, Mayur must have told that from Assam to all type of places, they are excited. I don't know, Mayur showed some slides of, they send messages at 2 a.m. in the morning. Yeah, so this is the type of thing that's happening across the country. So we are very excited because of the involvement, like a street cricket involvement. Street cricket you cannot stop. Yeah, you don't need a space. Anywhere they will start. I think Mayur must have stored that. So we are sure that science is going to be something like this if we remove it from its big aura. Yeah, so that's what we want to do.
Very nicely put. I think you made a very important point, starting from six Nobel Prizes on this beautiful organism. And why? Because it's a organism which is well studied, is a great model. A lot of people use it to understand biology and biological phenomena, but no one tried to, like you, to go take the knowledge to the kids and have them play with it at their homes and different places across the country. So you said that it instills scientific temperament, it instills curiosity, and that's why when they chat in your chat shala, they are discussing, and this is sustained. And this sustained interest in any organism, and especially Drosophila because it's very common, is the key to curiosity, is the key to knowledge advancement. And one can then go on and further characterize, study, screen different molecules, maybe anti-COVID drugs, and its immune profile, as you mentioned. So in order to captivate these kids and these college students into unraveling the beauty of a model organism, this is exactly what you're doing in the cube, which is so very exciting and inspiring for us. And this will take science further and should be evidence-based. And this is how the cube can expand to, maybe, as I once talked to you about, into schools, down not just to the colleges, further down to the high schools, and which will even, catching them younger, just like the cricketers are identified in different places in villages, and then they groom them, and then they grow as big cricketers. So similarly, if we instill this scientific commitment, this scientific curiosity and temperament at the kids level, the sooner they get to fiddle and to understand and remain curious at early in their lives, in high school maybe, and they would probably become good scientists. And the 7th Nobel Prize for the same model or some other model can come to our country. And when a Nobel Prize comes, obviously the translational value comes, because it may probably be used for human benefit or clinical benefit, I want to say. And the same applies to our audience here, who come from different background. There are people from the Ayush background who have some of them use models, some of them just use empirical case studies to make a claim for some particular drug or any of the herbal drug or siddha drug. But they must take it to the scientific exploration. They must document the evidence of how intervention leads to an improvement or no change, and whether at the human level, if it is ethical, or if one wants to look at the efficacy of a certain intervention, be it a drug, a herb, or it could even be yoga, then the blood products of having intervention can be tested in models, even in Drosophila or in mouse model, a mouse model of a disease, a genetic knockout model of Drosophila, or any organism where we can give a drug or give a blood product of a person who has undergone yoga or meditation, and then test if it rescues a certain phenotype, whether it has epigenetic effect. Dr. Arunan alluded to. He mentioned epigenetic basically means that there is an outside effect, outside gene effect, which influences gene expression. So this is a new concept which came up a decade back, and this is what he was trying to explain. And therefore, a model system is the core of scientific discovery and knowledge advancement. This is what he was trying to say. So although there are people who are doing their practices in yoga studios or in Ayush clinics or in modern labs, or in chemistry, synthetic analogs they are producing. But the most important thing is who is going to do this research. So he works on students so that to make them, mentor them as scientists who are full of enthusiasm for science. And obviously, when you talk of science, you talk of evidence. And that person who has scientific temperament will be able to distinguish or discriminate between subjective evidence and objective evidence. So he made a point for objectivity. And this Drosophila is a model to screen different biological phenomena using objectivity. And in order to captivate in the process of this objective experience, he uses Drosophila and Daphnia and et al., all these things. And the students are captivated and they enjoy growing it, understanding it, naming it in different states by different names. So that in one way or the other engages them in the process of scientific discovery. Did I correctly understand the message that you two gave?
Yeah. I just wanted to add one point probably,
Sure.
which is, or emphasize one point and add another half a point. So what we are you said about Ayush related questions. So what is important is the methodology that is to be used. So that's so you get a huge constituency of people who will insist on this methodological aspects. That's what you see, we don't have to talk about it. Students themselves will say that where is the evidence? School students
Correct.
will ask where is the evidence, that type of thing. So
Correct.
and then whether there is a control, they will ask what's the control. You have a test, but where is the control? Did you use a control type of? So this becomes a natural habit. And that's what I think we need to become, instead of just blindly believing something or blindly rejecting something. Both
Yes.
we do that very often. So
Correct.
that's one of the points that I wanted to connect with what you said about. The other I wanted to sort of slightly mention is that see, if you look around, we have not only fruit flies and this thing, we get several other things, and then we come to know that they are all interesting organisms which people have been working with, or maybe we can develop it as some model which with which we can address some questions which others were not able to address. Now, there is another organism called C. elegans, the small nematode, which is again Nobel Prize winning stuff. People have been getting Nobel Prize from 2000 onwards. So this is from in the soil, and we hardly look at these things. And so now in the home lab, they don't have a petri dish, so they are developing. I don't know whether Mayur said that or not. They are developing simulating a system like agar and petri dish. So they are boiled potato slices they are using in order to see that whether they can. So normally they isolate, extract the nematode from the soil through by using a petri dish with agar and stuff like that. So they are trying to do that. And so what I am trying to say is that anything you touch, anything that's a real interesting model or a question, which it can excite them because it is from their own doorstep. So it's almost
Sure.
a doorstep science.
Yes. And this may be happening now as audience from the interdisciplinary audience that we have must be listening to you, and they may want to know more how they can join your CUBE group and they do their kind of experiments, whether it's C. elegans or Drosophila. Can you maybe give an email, or this particular number is there? That is the WhatsApp number. Can they contact you there, or should you like to tell
WhatsApp number? They can use that. You want an email, and I think you must be having my email. I think can I give an email? Mayur,
Yeah.
can you just give the email?
Yeah, you can just speak out. Yeah.
Okay. mcarunan@gmail.com. mcarunan@gmail.com.
Arunan.
M for ARUNAN.
Here you go. This is Dr. Arunan's email ID, and that is his telephone number. It's kind of him to have spared so much time. Actually, he had to sacrifice his daily evening rendezvous with these students across the country online. And you can also join him, learn more about how to focus on developing knowledge for a model system or any scientific or any biological phenomena. So please note that down. Otherwise, we have this video recorded. You can always go back to our Facebook and look at the entire conversation. And you can also click pause and then jot it down if you haven't been able to note it down now. So Do you have any message to give to these young kids from different interdisciplinary area, or maybe their fathers and mothers who are watching? And then we can wrap it up.
I think they should join this very interesting group across the country, and some of them are also from outside the country. So I think they should join that, and I think it will. They won't regret. They won't regret. That's
I'm
what
sure. So thank you very much. You have been an inspiration to me also. Always, I derive a lot of inspiration from you, your talks, your enthusiasm, and your commitment to continued and sustained education. And thank you for bringing and introducing Mayur Gaikward to all of us. Prashant, do you want to ask something? I'm sorry I didn't give you an opportunity. Does any thought come to your mind?
No, sir.
Okay,
No.
okay. All right then.
Okay. Thank you very much. Okay.
Thank you very
Thanks a
much.
lot.
Bye.